course, about hemoglobin. I guess the one that shows up there best is maybe the green one. Is that the one that's showing up best for you? Let's just look at this green stuff. So how many of you read up and you know that this histidine residue right here, and if you point to it, it'll tell you what amino acid it is. How many of you read up in your textbook and found out that that heme group has a histidine that's proximal and distal? So that's the proximal histidine. That's the one that's involved in shifting that pushes the iron into the heme group. Because you remember reading about it, and I know you all want to now, because you're going to be like, what was Dr. Gibbons saying about that heme group? That was so cool. I want to go find out how it shifts into how the iron shifts in the heme group now. So you can come here and look at that, this residue, and that's the one that's proximal that shifts in there. But look at this one over here. This histidine residue here is pretty interesting. There's a histidine residue that's right in the way of, this is your O2, that's right there. So anybody ever wonder, have you been told that carbon monoxide binds to that iron in that heme group much more readily, has a much higher affinity for that heme group than oxygen? What does hemoglobin do?
Carries oxygen.
Carries oxygen, and if it doesn't carry, if we don't get enough oxygen carried by hemoglobin, we die. So it's kind of important that we understand why something like carbon monoxide that has a higher affinity, that'll knock you on the floor literally. Why doesn't carbon monoxide bond? And it's because of the primary sequence that's formed tertiary sequence that prevents that, because you've got a histidine residue right in the way. It forces the oxygen to bind at an angle, and oxygen is happy to bind at an angle, but guess what carbon monoxide is not happy with? Not happy to bind at an angle. And so this limits the carbon monoxide which has preferential binding from binding to that hemoglobin. So multiple things that you can do here, and I know what you guys are going to want to do later is go back and look at the sickle hemoglobin, right? Because anybody remember what's going on with sickle cell? What happened to hemoglobin? How many amino acids did we have changed? One, just one. Just one amino acid changed, and that protein no longer functions correctly, right? So one amino acid in the primary sequence changes, and now the tertiary sequence, quaternary sequence, isn't functional the way that it's supposed to be. That's profound. Anybody remember what that one amino acid is? It's a valine, right? It's been changed. So it's glutamate to a valine. And so the valine is what kind of how do we categorize valine? Hydrophobic, right? And so with that hydrophobic residue in there, and it causes aggregation with other hydrophobic regions. And so you saw when I opened this that there was a sickle of hemoglobin, so now you have something to do during dinner, right? Okay, so this is the last slide in the lecture, and this last slide in the lecture is showing you all of the instructions that I just told you about how to get to that protein database so that you can look up any proteins you want. And literally, you can look up any proteins that you want to look at in that database. Okay, scale all the way back. Alright, so I'm not going to introduce myself because you've already been introduced to me, but you have the pleasure of going over chapter 5, Techniques in Protein Chemistry, or I have the pleasure of going over this information with you today. So the key component that I want you to focus on when we talk about the proteome, and the proteome is literally all of the proteins in a cell under a given set of circumstances, under a given set of conditions. And we're going to focus on purification or isolation and purification of proteins. And then we're going to look at some chromatography techniques in addition to a few immunological techniques. And we're going to look at characterization of those proteins that were purified and make some inferences about structure and function and relationships. Now, we're going to be going over material very fast. Are you used to that? So, yeah? Okay, and so some of the slides I'm going to go over so quickly that you may want to refer to your textbook to get some more details, or you may want to form some study groups that you can talk to your classmates about, or alternatively talk to the TA or your instructor about some of the details. So the first thing we need to do is remove the proteins from the cell. We can do that by a process called differential centrifugation. We take tissues and we chop them all up into little bitty pieces. So we can take muscle tissue or we can take other organs, we chop it up, we put it into a tube or a blender, actually. You can put it into a blender, the kind that you use in your kitchen. And we're going to put a buffer. And that buffer will prevent the protease enzymes from chewing up our proteins. The buffer is also going to be some hypertonic, so it's going to lyse the cells and release everything that's inside of the cell. And then we're going to centrifuge for around 500 Gs at 10 minutes. We're going to pellet what's called this nuclear fraction. Those are the heavy nuclear components. We're also going to pellet unlike cells and some other cellular debris. We're going to get rid of that pellet, so we're going to discard the pellet part, we're going to save the soup, and then again, we're going to centrifuge that soup, this time around 10,000 Gs for 20 minutes and pellet out the mitochondrial fraction. Our proteins of interest, the soluble proteins in the cell are still in the supernatant, so we're going to discard this pellet. We're going to keep that soup fraction, and again, we're going to centrifuge. After we centrifuge now at much higher, 100,000 Gs maybe for an hour, we're going to pellet out the microsomal fraction, which includes all of the EPR, the plasma reticulum, other cellular debris, and any membranes that are still left in the cell. And we take the supernatant, and now we have soluble proteins that we can actually start to look at in terms of our protein of interest. So we want to pull out our protein of interest. We want to preserve the biological activity during the purification. Some of the techniques I'm going to talk about today won't completely preserve the biological activity. But we want to do this as often as we can as scientists are going to take advantage of differences in protein solubility, protein size, protein charge, and also binding affinity. One of the techniques we can use is called salting out. So proteins, and this is protein solubility over here, increasing from bottom to top. And this is the salt concentration increasing from left to right. Proteins need a little bit of salt in solution to be dissolved in the solution, and the reason for that is because they have positive and negative charges associated with the protein. And so they'll form a dipole ion interaction with the salt, and that puts them in the solution, and that's called salting in. But as we continue to increase the concentration of the salt, increase the concentration of the salt, turns out that the water will start to compete with the protein for the dipole ion interaction. And at some point, during the salt concentration increase, we'll salt the protein out. In other words, the protein will precipitate out of the solution. And it turns out that different proteins precipitate out at different concentrations of salt. And so we can, again, identify a range where our protein of interest might be. Then we want to get this all out, and we can do that with dialysis. And so we put our mixture, our protein mixture, the proteins are represented by these red spheres, and the salt by these little blue spheres. Put our protein mixture into dialysis tubing, which seals top to bottom. And by diffusion, the salt will diffuse out of the pores of dialysis tubing, which are too small for the protein to diffuse at all. And so now we have gotten most of the salt out of that solution. Mixture of protein. We can then put that mixture of protein on top of the gel filtration column. We use gel filtration chromatography, or also called side exclusion chromatography, to separate small and large molecules. So we put our protein mixture on top of this stationary phase, this column, and then we're going to run a buffer through that stationary phase, so our mobile phase is going to be buffered. And maintain our protein at specific pH, and also make sure that the protein doesn't get chewed up by any enzymes. Proteins are pretty nifty. When you take them out of the cell, they're not real happy. They like to be in the right temperature, in the right pH, and they like to be in the right environment where there aren't proteases. We run the buffer through the column and separate the proteins based on size. The reason is because we have carbohydrate beads in our solid state of this column. Those carbohydrate beads have tiny little pores in them. Those pores are small enough for these smaller molecules to enter the bead, but too small for these larger molecules to enter the bead. So what happens is the larger molecules will flow through the column unimpeded, and so they have a very short path length. So the smaller molecules have to take this path length going through all of these beads. And some of the intermediate molecules you can see will enter some of the beads as well. If we know the volume of this column and the volume inside of the beads, then we can take the total volume and the volume that's inside the beads into consideration. We can actually get a molecular weight for the proteins that you're eluting off of these gel filtration columns. So as we go through this, I want you to be thinking, compare and contrast these different types of chromatography. So how would this be limiting and how would this be really good? When we look at the next type of chromatography, it's ion exchange. And in this case, we have some synthetic or cellulose beads that are a solid phase in our column. And those beads can be bonded covalently to functional groups that are either negatively or positively charged. And so when we run our mixture of proteins through this column and buffer, the negatively charged, because the beads on the column, the stationary phase, are negatively charged, the proteins that are predominantly negatively charged are going to run through the column for the most part unimpeded. But those proteins that are predominantly positively charged are going to bond to those beads, those negatively charged beads. Okay? So how do we elute then our protein of interest that's bonded to this column? Well, we can either change the salt concentration, like we talked about a minute ago, or alternatively, we can adjust the pH. So let's just think about it for a minute. Let's just answer a couple of questions. So for a protein with an overall positive charge physiological pH, would you increase or decrease the pH to remove the protein from non-exchange column? What do you think? Do I still have all of you back there? Are you still with me? So what do you think? You want to increase? Why would you want to increase? So if you have a positively charged protein, overall positively charged, what kind of amino acids are associated with a protein like that? Basic. So give me a couple of basics. Lysine, arginine. Okay, so how do we get lysine or arginine to have an ideal electric sign? What do their charges have to look like? Yeah, zero or neutral. They have to look like zero or neutral. And so what would we do to get a species that has an overall positive charge, physiological pH, really? What would we do to that species to get the charges to be neutral? What group do we have to affect? Because you know that the carboxyl group has what kind of charge on it? Yeah, the carboxylate group is negatively charged at some pH 2, right? And so what other group do we have to pull a proton off of?
NH3.
Yeah, maybe the alpha amino group, because arginine and lysine, there are groups have a higher pKa, right? And so if we can pull a proton off of that alpha amino group, now it's not positively charged anymore, it is neutral. The R group is still positive, and the carboxylate group is negative, and so it is neutral. And so raising the pH, and I have one more question for you. Would you expect more or less of sample loss from this technique, that is ion exchange from phosphate, versus gel filtration? What do you think? So let me ask you about the interactions. Are there interactions with ion exchange chromatography? Are there interactions of some sort with column? What kind of interaction? What kind? Electrostatic, right? We've got positive and negative charges that are associated with that. What about gel filtration? Any interactions with the column? Any bonding type of association interactions? Attractive forces. There's what? Yeah, they're neutral, right? So they run through the column, they just have to go in and out of the beads, but they're never actually adhered to the stationary phase, right? And so I'm going to show you another method called affinity chromatography, and I want you to think about these three and think about which are going to hold more of my protein, because during the purification process we use all of these and we lose our protein at a certain point. And so the question is, where am I losing the most of it? So this technique is called affinity chromatography. And again, we have cellulose or synthetic beads, but this time they're covalently bonded not to just a positive or negatively charged functional group, but to a specific molecule. And that specific molecule, in this case glucose, has a binding affinity to our protein of interest. And in this case, in this column, our protein of interest is continatin A, which binds glucose very specifically. So we add our mixture of proteins to this column. When we add our mixture of proteins to this column, specifically continatin A, it's going to associate with our stationary base because it's going to bind to glucose. All the other proteins are going to do what? Run through unimpeded. All the other proteins are going to go through unimpeded. And so then how do we release those species? Well, we add a high concentration in our blood or in our blood of glucose. And we set up a competition for continatin A to bind to our mobile phase versus the stationary phase. And so what happens is our continatin A will be in the stationary phase, start binding to the mobile phase that's moving through because the glucose is in a very, very high concentration. Okay, so properly bind. Do you think we lose some of our protein of interest on this column? What do you think? You think we get all of it off of there? You think some of our continatin A is going to say, you know what, I really don't like red glucose. I like green glucose. Bang. You may not no red glucose. So we'll probably lose some of our protein of interest because some of it is going to stay bonded to that column. So scientists don't like to wait. Do any of you like to wait? So when you put a website onto the computer, you want to come up right away, right? That's how I understand. And so what we found is that with the types of chromatography, the methods I've shown you so far, is that they just don't have the resolving power we want. And so if we want better resolving power, we have to get very fine beads so that we can have more of what? Interactions between the beads and our protein that we're running through the column. But the problem with that is because that does improve the resolving power, but what happens to the flow rate? Now we have more stuff packed in the column, and so the flow rate is very slow. So we remedied that with high pressure chromatography. This is high pressure liquid chromatography. There's other types, high pressure gas chromatography. We remedied that by hooking a pump up. And so here's our mobile phase, and we're going to literally pump our mobile phase through that column. It hits a detector, then it goes through much faster, so now we're eluting pretty quickly. It's a detector, and that detector will give us a display of our proteins as they come off the column at 220 nanometers because the carbonyl group is double bond O. Double bond absorbs at 220 nanometers. So we have a good way to detect it. So if this data over here is representing gel filtration, HPLC, how does gel filtration separate proteins? Based on what?
Polarity.
Based on what?
Polarity.
Based on size. Based on size. Good, just see the back is wide away. Somebody over here. All right, so based on size, gel filtration separates proteins based on size. And so what I'm asking you is, even if you don't know what these proteins are, you don't even know the identity, you're looking at the solution profile, what can you tell me about protein 1 versus protein 5? The biggest. And you know that because it came off the column when? First. It came off the column first, and so what came off the column last is the smallest, and 2, 3, and 4 are somewhere in between that. You're already gel filtration expert. We got it. So let's talk about another method of separating proteins of SDS case, that is the decyl sulfate polyacrylamide gel electrophoresis. And this is SDS right here, and you can see that SDS has a long hydrocarbon chain and then this very polar head group up there, the negative charge on sulfate, right? So this kind of looks like detergent. That looks like a soap, right? Long hydrocarbon chain and then the polar head group on it. And it is actually a detergent, and what it does, so here's a protein before SDS. So we have positively charged group, and SDS, this negatively charged SDS, is going to react with those positively charged groups. And then, because we have this long hydrophobic tail, we can disrupt all of these hydrophobic interactions as well. Another thing we put into SPSH is beta-mercaptoethanol, which breaks all of the disulfide bonds, reducing the. So essentially, we linearize the protein and we put a negative charge all the way around it. So how will we be separating using this method then? If all of the proteins are basically the same shape, then they're going to separate based on size. They're going to separate based on size as well. There's a problem with this technique. So if we're denaturing this, and we're disrupting all of the disulfide bonds, then what protein structure are we going to lose? Quaternary. We're going to lose quaternary structure. And so that's one of the problems with SDS. We're going to lose quaternary structure. And we could separate by size through gel filtration, and that doesn't happen, right? So we put a polyacrylamide gel between two electrodes, negative and positive electrodes, and we load our protein mixture onto that gel and hook this up to an electrical current. And literally, the current is going to pull the proteins through the gel pore, the polyacrylamide gel pore. So which one do you think is going to hit the bottom first in this case? The smallest one. It's going to pull the smallest ones through the gel first. Okay. And they're going to get sequentially larger as we get to the top. The very largest proteins are going to be at the top of the gel, and the very smallest are going to be at the bottom of the gel, and all the sequential are going to be complete. We can visualize that gel after we run it, and you saw it was a clear polyacrylamide gel, basically colorless. We can stain it with Kumasi's blue dye. And this is 3 different mixtures of proteins, so that we can visualize all of our proteins where they are at. And if we run a standard, what we call a standard, on this gel, which is other protein mixture, then we can compare the bands on the standard to the bands on our independent lanes of our protein mixture and make determinations about the size of those proteins. So this up top, you guys have seen before in your chapter 3, I think, and so you know that this species is called what? It's zwitterion. And it's called zwitterion because it is neutral, basically a pH. And so, but this species has an R group that is also neutral, right? So we're talking about a neutral R group. When we talk about proteins, we talk about having a dominant positive or dominant negative charge associated with them. And so what if, for example, this R group right here had a positive charge associated with it? We talked about this 50 slides back, right? And we said that then this, so what's the overall charge right here now at physiological pH? Positive, overall positive charge on that species right now at physiological pH. And so in order to get an isoelectric point, and that's what isoelectric focusing takes advantage of, the isoelectric point. Okay? So we're going to put our protein mixture into a pH gradient that has a charge associated with it, positive and negative. Where would a species like this head toward in a pH gradient? Toward the positive pole or the negative pole? It's going to head toward the negative pole because it's overall positively charged. When is it going to stop? When will it stop between those two poles, positive and negative charge? It's going to stop when it's what? Neutral. I heard somebody say it, but this name's quiet. So neutral. It's going to stop when it's neutral. And so is this species neutral? Yeah, there is a negative charge here and a positive charge here, but what happened to the alpha amino group? Deprotonated it. And so, for example, here we have overall positive charge, move towards the negative pole. Isn't that what you told me it would do a minute ago? You're not so sure anymore? That's what you said. Okay, so it's going to move towards the negative pole until it hits its isoelectric point, where that protein is neutral, and then it's going to stop. And that's the way we can separate protein mixtures using isoelectric focusing. But there's a drawback. There's a drawback because each one of these bands that we have down here represents multiple proteins because there are lots of different proteins that share the same isoelectric form as well. It's not like separating individual amino acids anymore, it's a little more complicated than that. So what we can do is attach this polyacrylamide gel from the isoelectric focusing to another polyacrylamide gel, and then we can run SDSK in the opposite direction that we ran the isoelectric focusing. And so now we can resolve the proteins that are associated with this band, and we can resolve the proteins that are associated with this band, proteins associated with this band. So is this one small or big? It's small, right? It's down to the bottom of the gel. And if you look at a real gel over here, or a photo of a real gel, figure of a real gel, here's our isoelectric focusing up here. Look at this big bunch of proteins that all have a similar isoelectric. And so then when we run SDSK in the opposite direction, we resolve a lot of those proteins. Now, same thing with this big bunch of proteins right here. Resolve a lot of those proteins. In fact, we start out with these major bands, and we end up with all of these proteins that were in our mixture. Protein purifications can be monitored. We can assay the activity of the specific activity throughout the purification process in the case of an enzyme purification. We want to monitor the specific activity, which is the ratio of the specific of the enzyme activity to the protein concentration. And we can measure the effectiveness of the purification scheme at every step. For example, if we had lactate dehydrogenase, this is a convenient assay because NAD plus is reduced to NADH as lactate is oxidized to pyruvate. And NADH is really convenient to measure because it has a spectrophotometric absorbance at 340 nanometers. And so we literally can detect whether or not NADH is in the system, whether or not lactate dehydrogenase is catalyzing an enzymatic reaction. We can keep track. And so this table is keeping track after every step. Homogenization, salt fractionation, ion exchange chromatography, gel filtration, and affinity chromatography. And you notice as our total milligrams of protein decreases as the purification process is going forward, our total activity decreases, which makes sense, right? We have less protein, we have less activity at each step. But look at our specific activity increasing over here from 10 to 30,000. And so that specific activity, again, is a ratio, right? It's a ratio of the total activity to the total protein. So that's why it's increasing, because we're enriching our enzyme that's in solution. I'm going to ask you a couple questions about that. Purification level at affinity chromatography, yeah, 30,000 is the specific activity units per mg. And 3,000 is the purification level, which is the specific activity at each step. So we look at this, there's 30, that's the specific activity at each step divided by the starting specific activity. Great purification. And we can visualize this, so not only quantitatively, but qualitatively, because we can run those proteins at each step, the homogenase, the salt fractionation, the ion exchange, gel filtration, affinity chromatography. We can run our protein mixture at each step and make determinations about whether or not we're enriching our protein of interest. And you can see it after affinity chromatography. So we only lose a couple between homogenase and salt fractionation. We just lose a couple bands. We only lose a couple more on ion exchange, because we still have a lot of proteins in our mixture, a lot. And then with gel filtration chromatography, we isolate the fraction of those proteins that are around the size of the protein that we're looking for, and then affinity chromatography leaves us with a highly enriched sample. So why do we do an assay after every step? Doesn't that seem kind of laborious? Do we have to do an assay after every single step, really? Why do we do that? I mean, somehow that's the case. Assay the activity and then the specific activity, and we got to get the purification factor and all of that. Can we skip it? It's like skip 3. So we skip 3. And we find out after we've run 3 different types of chromatography, guess what we don't have when we finally decide to have it? Our protein of interest is gone. We don't have it. And so now we've wasted a lot of time. And so at every step, you want to make sure that you have your protein of interest. And then I want to ask you a question based on the purification scheme shown in table 5, and I'll back up in a minute. Affinity chromatography appears to result in the highest specific activity and purification level, so doesn't it seem more straightforward to just dump the rest of them? Can't we just go straight to? So there's the gel, the affinity chromatography, there's our protein, completely in red. And here's our purification scheme quantitatively. Why don't we just, look at this. This is great right here. Why don't we just skip all this stuff? And just do this technique, affinity chromatography, pour our homogenate on an affinity column. Purify it. Because it what? Okay, so that's a good point. She said that there's only like 1.75 milligrams of that particular protein, the one we're interested in. So what's the problem with that?
Low yield.
Okay, low yield. And what's the problem with just running something like that on an affinity column? What is it? Yeah, you might not even see it. And so that's like you getting to a concert that's packed with people and trying to make your way up to the front, right? So there's proteins everywhere. We have this high concentration. So look at the gel again. We have this hugely high concentration of proteins on this gel. And our protein of interest, this is one little 1.75 mg. And so how are we going to get it to bind to a solid phase column with all these other ones in the way? We're going to lose a lot of it. And so we may lose most, almost all of it, or so much of it that we never have enough to assay. So we can't just cut to the chase. So some proteins can't be purified solely by the techniques that we just discussed. And for example, the estrogen receptor has no enzymatic activity, and so we can't keep checking the activity at every step. There are immunological techniques that take advantage of binding, specific binding. For example, a protein receptor codes ligand or an antibody to an antigen. And we're still going to use centrifugation, in this case ultracentrifugation, because it's still a really good method to separate proteins. In terms of ultracentrifugation, which the centrifugal force is described by this formula right here, where the N is mass. And this is the buoyancy factor, this mu symbol with the line over it, that's the reciprocal of the particle density, and the rho symbol is the medium density. And then this lowercase italicized f, that's the frictional factor. And so we have to take into consideration all of these components because proteins have different masses, right? But they also have different shapes. And so if we had a really compact protein that had the same mass as another protein that was more widely dispersed, then which one do you think is going to be able to penetrate into the centrifugal force more? Compact one, or if it were more dense, right? And so we take all of these components into consideration when we consider how far into the medium is our particle going to travel. And turns out a lot of people have put together values associated with that. So the S value, which is given in February units, tells us how far, essentially, a particle or a protein, these are all proteins over here, is going to travel into a medium under centrifugal force. And we notice that as we increase the molecular weight, what happens to the S value? Sedimentation. Okay, you guys, this isn't a trick question. Really,
Increases.
I'm up here.
It increases.
As we increase the molecular weight, what happens to the S value?
It increases.
It increases. The S value increases, right? So, but there's a glitch. There's always a glitch. That's always the one they put on the test. Remember the straightforward stuff, the glitches, right? And I'm not saying that's a tough question. I'm not your major PI, but I'm just saying you should pay attention to the glitches. So 12,310 has a greater S value than 13,690. Why could that be? Yeah, so we could have more compact structure or denser, right? So this cytochrome C essentially, which has lower molecular weight, but higher value over here, because it's more compact, it has more dense structure relative to the ribonate. We can amp this up in terms of resolution by getting a gradient. So we can get a sucrose gradient. We have a low density, maybe 5%, maybe 20% over here, and we drain that into a centrifuge tube, and at the bottom of the tube we're going to have denser sucrose, and we will at the top of the tube, we lay our sample there in the centrifuge. And when we centrifuge, we're going to separate those particles based on their sedimentation coefficients. And you see there's still a black line up here because some of those species in that mixture may not be able to penetrate the medium that we have. We can poke a hole in the bottom and separate into fractions. And we can use this for the estradiol receptor complex. When we talk about the estrogen receptor binds to estradiol fairly tightly, we can use this to determine whether or not we have the complex. We incubate the mixture, the protein mixture with radioactive estradiol, and that estradiol is going to preferentially bind to the estrogen receptor. The unbound steroid is going to be too small to migrate into the gradient, and so what we detect radioactivity for is going to be our complex estrogen receptor with the estradiol. So when we look at that, in terms of disintegrations per minute, the radioactivity increasing from bottom to top, you can see that our steroid only didn't penetrate. And so we have lined up here all of our fractions that we took from the bottom of the tube that we bring up to the top of the tube, and at the top of the tube are the species that don't what? Don't penetrate the medium, right? So we read the disintegrations per minute as we go from one fraction to the next, and we find that we have a peak here at 4x. And so we know from this that our estradiol estrogen receptor complex are definitely there, but there's a problem with taking into consideration that we can just isolate this fraction right here, and that's going to be our estrogen receptor estradiol complex. What's the problem with that? So I gave you a hint. It's right there.
Proteins.
Yeah, there's other proteins that are going to have a sedimentation coefficient of 4. And so in this particular test tube, we have more than just our protein of interest. And so we're going to have to separate it using another technique. And one of the things that we can take advantage of is the antibody-antigen binding interaction at the epitope or the antigenic determinant, because it's very, very specific. And so, for example, if we set it up so that our antigen were the estrogen receptor, and we've got a monoclonal antibody for that estrogen receptor, then we could essentially fish it out of that mixture in our test tube. So that's what we do. We have our monoclonal antibody over here, and it is covalently bonded to a bead. This could be a synthetic, probably synthetic or cellular bead. And then we pick cytosol for maybe rat uterine. We want something that is enriched with the protein that we're after. And rat uterine cytosol is enriched with the estrogen receptor. We mix those together and we centrifuge. And the heavy particles are all going to repel. All the heavy particles. That's going to be our bead with the antibody bonded to the bead covalently. And what's going to be bonded to the antibody? Remember, the antibody is specific for the estrogen receptor, right? And so it'll be the estrogen receptor that's bonded to it. And then we discard the supernatant, we save the color because that's where our protein of interest is, wash out all of the extra proteins that we don't want here, and then we add denaturing. When we add denaturing, what is that going to do? Disrupt what? The interaction between the antibody and the estrogen receptor. And so what kind of bonding interactions do you think those are? If we're adding denaturing to disrupt the attractive forces between those two components, do you think they're covalently bonded together? Do antibodies and antigens bond together and with covalent bonds? So what does the denaturing do? What kind of interactions does the denaturing disrupt? Hydrogen bonds, which are a strong form of dipole-dipole. some hydrophobic interactions, and so van der Waals, and so those are the kind of interactions, attractive forces that we have between the antibody and the antigen. There are other methods. This one, ELISA. The ELISA is an enzyme-linked immunoassay. And so we can do high throughput with an assay like this because we can do this on a 96 well plate. And so a lot of times they use these types of assays to screen for something. We bond either the antigen or the antibody to the well plate. And then to that antigen will bond a specific antibody, or to this antibody and sandwich ELISA will bond the antigen. So in direct, we start with the antigen, sandwich, we start with the antibody. We can commercially purchase this antibody that is specific with an enzyme, like, for example, horseradish peroxidase, which undergoes a color change when you add substrate to it. So that enzyme catalyzes the reaction that undergoes a color change when you add substrate, and that enzyme is bonded to this antibody. It's bonded to either our antigen or our antibody. When we get a color change, then we know that either our antibody is present or our antigen is present. And you can even take the color change and calculate the potential concentrations that you have of those specific solutions, depending on the color. Another way that we can use the antibody, antigen, specificity and binding is with Western blotting. It's also called immune blotting. We run an SES page, separate our proteins, then we transfer those proteins to a polymer sheet. We can't run the antibody in the SES page to start with because its mass is different from our proteins of interest. So we separate our proteins of interest, transfer them to a polymer sheet, and then add a fluorescently labeled antibody and incubate that. And then we wash any excess antibody away that hasn't bonded. And then we can illuminate this gel, this polymer, this immunoblock, not the gel, but the polymer sheet. We can illuminate it with light, and we see our fluorescent emission. And our fluorescent emission is going to occur right with the same protein that our fluorescently labeled antibody was specific for. And if we run that with a standard, we can tell the the size, right? Yeah. But some of you, I know, are answering me. I can't hear you because it's loud up here. All right. So the primary protein structure is key in understanding protein function, and one of the preliminary steps that we do is determine the amino acid composition of the protein. We're going to hydrolyze every peptide bond in the protein so we can separate all the amino acids. And then we're going to visualize them with a dye, fluorescent, on an ion exchange column. Here's the dye, and you notice that this oxygen binds to the alpha amino group, and nitrogen is the alpha amino group. And so now we're going to get a fluorescent signal. We dilute our protein mixture, and then we can compare our experimental solution of our protein mixture with a standard amino acid solution profile. And we can make determinations about what the amino acids are this way. So we have arginine, phenylalanine, alanine, glycine, and starches. So now we determine the composition, so we pretty much know what amino acids are present, but we don't know what sequence those amino acids are in. And so we can use phenyl isothiocyanate. This carbon is going to bond to this nitrogen, forming a carbon-nitrogen bond that you see right here. And then this pi electrons are going to go back and attack that carbon on this carbonyl, forming a cyclic structure and breaking this peptide bond right here. Essentially what we do with Edmond degradation sequencing is we break one amino acid after another off of the N-terminus. After we get this cyclic structure, we have to run these species through ion exchange to identify what amino acid that is. It's pretty laborious because every time we run Edmond degradation, we get another cyclic derivative of amino acid. We have to identify it through ion exchange. We can only do this up to about a 50 amino acid protein, and really only about 30 amino acid protein. So what if we have a big protein? What if we don't have a little peptide that's 50 amino acids long, or 30 amino acids long? Then we'll undergo cleavage, and we have different chemical reagents and different enzymes that will do this cleavage, and let's focus in on only two, trypsin and chymotrypsin. The trypsin and chymotrypsin both cleave at carboxyl sides, but on different residues. So based on cleavage at arginine and lysine to trypsin, or alternatively, tyrosine, tryptophan, phenylalanine is the green amino acid structure, or leucine and methionine. We can digest our protein with trypsin, and then run enzyme degradation and get two small fragments. We digest the chymotrypsin, and then run enzyme degradation and get three small fragments. And overlapping of these fragments with the fragments by chymotrypsin will give us the sequence of the amino acids. Okay, so this is just giving you a list of all of the things that understanding the primary structure can facilitate, and ultimately we're interested in purification, characterization, and the structure-function relationships because it's different about primary structure after we do that. Any of you that are remaining have any questions? Yeah. Okay, so you're all solid on protein, purification, characterization, and primary structure. Great.